And to really reinforce this and add to the complexity, if we take a point in time, not all microbiomes are equal. This is a study of the gut microbiome in developing and developed nations, in adults in this case. Here in red and green, we have gut microbiomes of Malawian and Amerindian populations. In blue, we have the US population. And basically, each spot is a profile of what type of bacteria were in the gut microbiome of these individuals. And how we work through this immense amount of data that we generate is we ask how similar is microbiome profile A to all of the other microbiome profiles in our cohort. And we calculate a distance. How similar is it? How close is it in terms of which microbes are there and what relative abundance, how much of them are there? And that's just a visualization of this distance calculation. So if we have two spots representing two gut microbiomes of individuals in these studies that are closely plotted beside one another, it means that those two gut microbiomes are very similar to one another. But what you can starkly see in this is the US gut microbiomes are very different from those of the Amerindian and the Malawian population. And even though the Malawian and the Amerindian populations are on two different continents, their gut microbiomes in these less developed nations are more like each other than they are like a US population gut microbiome. To reinforce what we've done to our microbiomes in the US, this is just looking at the number of types of bacteria that are detected across these populations. We have severely reduced the breadth of diversity and the number of different types of bacteria in the US population compared to those in less developed nations. And the really wonderful thing about this study is we got indications why this might be happening. When the study examined with shotgun metagenomics, looking at all the genes and the pathways in these microbiomes, what really differed between these populations, what was really striking is that the adult population of Amerindian and Malawians were really enriched for alpha amylases. So this is an enzyme that breaks down complex plant polysaccharides. So the diet in Malawi and in the Amerindian population is predominantly a plant polysaccharide, a plant-based diet. In the US population, we see huge enrichment of microbial metabolic pathways for processing simple sugars, found in processed foods, as we all know. So at least one feature, one thing that we know is driving these differences in the gut microbiome across these populations is differences in diet, in what we consume. And this was reinforced even more recently by Pete Turnbaugh, who is a faculty member here. This was a really wonderful diet-based study of 10 individuals. And what Peter did was he took those individuals and looked at how a plant-based or an animal-based diet may actually influence a healthy gut microbiome. And here we're just showing you the amount of fiber in their diet of these individuals before they started the study. Then they got, 4 days of a plant-based diet, or the other participants got 4 days of an animal-based diet. And as you would expect, the fiber content with the plant-based diet, or the plant polysaccharide content, goes up quite high. In an animal-based diet, it's very low. Also, the fat intake is lower in the plant-based diet compared to the animal-based diet, and the protein content is also dramatically different across these two diets. And he could track that these key dietary components really shift with the introduction of either a plant-based diet or an animal-based diet. What was really striking is that when Peter calculated, again, the distance, how similar are the microbiomes of the individuals after they start their plant-based diet compared to before they started their plant-based diet? Didn't really see much in the way of change. Plant-based diet doesn't really perturb the gut microbiome. However, in comparison, the animal-based diet introduction really increase this distance. And what that tells us is that microbiome is very different from the microbiome that was there before the introduction of the plant-based diet. But it's not just about changing the composition of the microbiome that matters. What the study also showed is that you change the molecular output of the microbiome by changing the diet. And here we can see that two key short-chain fatty acids, acetate and butyrate, were significantly reduced in the animal-based diet versus the plant-based diet. And that makes sense because these are the products of microbial fermentation of plants, of fiber. And what's really critical is these short-chain fatty acids are crucial energy sources for the cells that line the gut. They're anti-proliferative and they're anti-inflammatory. And we think they've become these, they have these activities because we've co-evolved with these microbes who traditionally have fermented our plant-based diet into these small molecules which quench inflammation and promote kind of health in the system. And so based on this, I'm sure you're not going to be surprised that we're finding an ever-increasing range of diseases are related to perturbations to the microbiome. And things, like the skin microbiome is perturbed in dermatological conditions, like psoriasis, for example. But what's really exciting is that we are now seeing that conditions like obesity is also linked to gut microbiome perturbation. But what's most exciting for me is that we're finding that conditions that are very difficult to treat and that we really don't have a handle on, like depression and autism spectrum disorder, are also linked to perturbations in the gut microbiome, suggesting that the gut microbiome may actually influence remote organs. And there's a couple of really key seminal studies that have shown this. They've shown that perturbations in the gut microbiome are associated with autism spectrum disorder and also with cardiovascular disease. But importantly, what these studies have shown is that it's microbial metabolites, microbial products that are responsible for these disorders. And much of this work has been done in mice with some follow-up work in humans. So it suggests that the gut is not like Vegas. What happens in the gut doesn't stay in the gut. It actually enters the circulation, and these small molecules, and perhaps there's some inklings even microbes themselves, may actually translocate to other sites across the body and change the physiology of the organs there, contributing to the health or disease of those remote organs. But how do we really know that it's the gut microbiome that's responsible for this? Well, that evidence has come from some really elegant mouse studies. So in these studies, in this case that I'm showing you, the feces of obese individuals or lean individuals were transferred into germ-free mice. These are mice that have no existing microbiome. They're bred to lack a microbiome. They're not particularly healthy mice, but they're bred to not have a microbiome. They're a wonderful vessel for studying how microbial introduction into kind of a pristine environment may shape the physiology of the host. And that's what these studies have shown us. Transfer of the obese microbiome into a germ-free mouse sets up an obesogenic microbiome in that animal. And those animals gain weight at a much faster rate than that of animals who received the lean microbiome, suggesting that the phenotype of the disease can be transferred from the patient by transferring the gut microbiome of that patient to a mouse. That's pretty incredible. That means that the microbiome is responsible in large part for obesity in this case. What's exciting is that it's not just obesity. This has also been shown for kwashiorkor. This is a wasting disease with neurological deficits that is really, can be quite prevalent in underdeveloped nations like Bangladesh, for example. Same thing. Transfer of the kwashiorkor gut microbiome or feces to germ-free mice induces wasting disease in those animals. More recently, it's actually been shown for autism spectrum disorder. Feces from patients with ASD transferred into germ-free mice induced neurobehavior that is consistent with the symptomology of the disease. So again, we're finding multiple disease indications where we can recapitulate features of the disease in a mouse who receives the microbiome from the patients with the disorder. So what can we do? Well, we call it yellow soup for the soul. Fecal microbial transplant. I'm sure you've all heard of it. It's not new. I call it yellow soup for the soul because there are records in 5th century Chinese medicine of producing yellow soup from feces as a treatment for gastrointestinal conditions. We've just recently rediscovered fecal microbial transplant. And what's very exciting is that it is essentially doing what we do in the mice, but instead it's transferring the healthy gut microbiome from a healthy donor to the gut microbiome of a patient with a disease or a condition or an infection to try and reconstitute the gut microbiome of the patient and treat the disease. So this has 92% efficacy in patients with Clostridium difficile infection. Antimicrobial treatment for Clostridium difficile is about 30% effective. And in this trial that used fecal microbial transplant to treat Clostridium difficile infection, they actually stopped the trial early because it was really they couldn't not treat the patients with this treatment because they were seeing 92% efficacy versus 30% in the vancomycin taper antimicrobial treated patients. It was unethical to continue the trial and not use this to treat patients. And we actually offer this at UCSF as a treatment for Clostridium difficile infection. It's also been used in a small pilot early study of children with autism spectrum disorder. It's about 18 or 19 children in the study. There they saw significant reductions in neurobehavioral symptomology in those children. In this case, instead of a single treatment where there's a colonoscopic delivery of the fecal slurry into the diseased gut, they did that initially, but then they followed it up with a month of sustained microbial pressure in which the children actually consumed freeze-dried fecal capsules. And that was sufficient, a month of treatment, to significantly reduce the neurobehavioral deficiencies in these patients. They've also recently followed up 2 years later with these children, and this effect is sustained. And in fact, they've seen even greater improvements across this small cohort that has been treated. And so now this is under clinical trial in much larger placebo-controlled studies across the country as a potential treatment for autism spectrum disorder. We at UCSF have been looking at inflammatory bowel disease. I'm in the gastroenterology division. I can tell you that we looked at Crohn's disease and ulcerative colitis, and fecal microbial transplant does not work for Crohn's disease, at least the way that we tried it with a single colonoscopic delivery. We had several adverse events and we shut down the study. And I think it's important for that message to get out equally as the 92% Clostridium difficile efficacy message. It's not equal. The microbiome is not the answer to all of our patients' ailments. And I think that it's important that we're cautious and that we are careful about how we implement this field and how we use this field to treat our patients. However, with an approach very similar to that taken in the trial of autism spectrum disorder patients, we're now at about 40% response rate in our ulcerative colitis patients. And that's really exciting. You know, our biologics are about maybe 20% efficacy. And we try different biologics in patients to ask what will work for them. But we're seeing 40% response rate with fecal microbial transplant. And we've got ideas how we can even enhance this even more. So for me, we're at a watershed moment in human biology. We've just discovered that we have this ancillary microbial genome that really influences our health, that shapes how our cells work and influences our health status.